Hi everyone, hope you're doing okay. This is our first lesson. So lesson number 1 is going to be microscopes, part of our cells, tissues and organs module. So let's get straight into it. Okay, so quick overview of what we want to look at today. I'm going to start us off looking at some careers to do with cells or biology in general, really. It's always good to get a little bit of background in where all of this knowledge could lead one day. Then we're going to label the microscope. Okay, so we're going to look at a basic microscope and label the key components. Describe how to use it, which is really important. Would be important a little bit further down your school journey. And then a little bit of calculations, okay? Calculating magnification. So it'd be really handy if you had some paper and a pencil or a pen to hand, and then you can join in as we go along. Okay, well, here's a happy guy. So jobs that use knowledge of biological systems and processes. First of all, what do you think? Can you name this job? Stethoscope, white coat, looking very serious. That's some notes. Yeah, almost certainly a doctor. What about this guy? Okay, this is close to a job I used to do. So he's watching Netflix, holding up a little beaker of water. I'm sure he's not watching Netflix. Okay, yeah, that's a water microbiologist. Okay, or I'd accept biologist or microbiologist. Okay, here we go. What about this one? Okay, this could be a job you end up doing if you follow a biological route. Okay, you've been in a building, you've been in a lab. There's some cultures there. This would probably be a research scientist, okay? Someone looking at cells, perhaps here, some HeLa cells, doing experiments to see what happens. So this guy would definitely need a microscope. Okay, what about this? What's he doing? Is he attacking him? Well, it's a very serious job, this one. Okay, so there's some clues in the background. You can see a spine. Okay, some ligaments and tendon models. Okay, so this is a physiotherapist. You've got to know a lot about the human body to be a physiotherapist. All right, okay, so some microscopic images. Okay, this is just to test you. I don't expect you to really get any of these, but what do you think this could possibly be? This is something that's been magnified. Okay, so this actually is a magnified view of a bird feather, which is really interesting because you can tell by the shapes there of the fibers in the wing, in the feather. They almost look like mini wings, okay? And you can see how airplanes get their shape for their wings. Very aerodynamic. Okay, what about this one? And I've told you straight away, haven't I? It's a butterfly wing. Okay, so this is a butterfly wing, magnified greatly. I think I'm probably going to have to just tell you the next one. This is a soft tick, okay? Don't get too freaked out by that, but that's what they look like when they've been magnified. A tick, obviously, you can find these out in the countryside. This is a body louse, okay? A little bit like a nit, a little bit like a head louse, but different. This one doesn't live on the body, despite the name. This one lives in fibers, so this would live in your clothes and lay its eggs in your clothes, and then it would only come onto your body to feed. Oof. Okay, but that's what that looks like under the microscope. It's a different world. One of my favorites. Okay, tardigrade. Here we go. Look, tardigrade. There he is. Okay, water bear. I used to look at these down the microscope an awful lot when I was a water scientist. So these are very, very sturdy little organisms. Okay, they can survive an awful lot. A lot of temperature changes and all sorts. Very resilient. And they look like little bears. Okay, so let's get on to looking at the microscope. The only way it's possible to see any of these organisms or any parts of these organisms would be using a microscope. The first part we will have a look at here, this is the stage. Okay, the stage is where you place anything you want to look at. It's quite maybe a straightforward one to remember because it's like a stage in a theater. Okay, it's flat and it's just something to put something on. Okay, so we call that the stage. Next, we have the eyepiece lens. The eyepiece lens is a lens, as it suggests. Okay, and it's what you look down using. Guess what? Anyone? Your eye. Yeah, so your eyepiece lens. This is the one you look down. And then the objective lens. Now, this is a lens that looks at the object. Okay, so it's the objective lens. So you've got two lenses, one at the top, the eyepiece lens, which you look down, and the objective lens, one which is close to the object that you want to look at. And that's the one you can change. Okay, we'll get onto that, but you can change the objective lens to magnify it even more. Okay, to look closer. Okay, so I'd like you to look at these, okay, and match the following keywords to their definitions. Okay, I'll just give you a minute to have a go at that. You can do this in your head, or you can write it down. I'm going to have a drink whilst you do that. Okay, so remember we've got the stage, the objective lens, and the eyepiece lens. Okay, let's look at these answers. So hopefully that not too many things to catch out there. The stage is where the slide is clipped down. That's just where we put the slide, the thing that we're going to look at. The eyepiece lens, where the viewer looks through to see the specimen. So the language is slightly different now. So the viewer looks through to see the specimen. The specimen is just the object that we put on the microscope to look at. And the objective lens is the part that changes the magnification of the image. So the objective lens is that one closest to the object that you can turn around and you can change the magnification. So the part that changes the magnification of the image. Okay. So if you want to put in the comments, feel free, by the way. If you want to put anything in the comments, say how you did on that one. Did you get 3 out of 3? How are you feeling about it so far? Okay. So let's look at another 3 pieces of this microscope. Right underneath the microscope is a mirror, and the mirror reflects light back up through the slide, okay, the specimen that you've got on the microscope, and that produces light from behind the image so that you can see it. Without that, you wouldn't see anything because it would just be dark. Okay, you'd just be magnifying darkness. So we need this mirror to reflect light up onto the specimen, and it's exactly what it says it is. There's nothing too fancy. It's a mirror. It's a piece of reflective glass. Next, we have the fine focus. The fine focus is at the bottom of the microscope. Okay, you've got 2 knobs. You've got your fine focus at the bottom. We'll get onto the second one in a moment. The fine focus is fine. Okay, so it's very small increments, small changes with the fine focus. Okay, as the name suggests. Okay, it's very fine. You're not making big changes. You're making very small changes to the focus here. Very small increments. And then, of course, you've got the coarse focus. The coarse focus is at the top, and this one moves it in much larger increments. Okay, so this is when you're trying to initially find something on the lens before you want to get really close into it using the fine focus. You would use the coarse focus. The coarse focus moves it much quicker, and it enables you to find what you're looking for initially. And then you would use your fine focus to get it into focus. Okay, same again. I'll give you another minute. Have a read through these and see if you can match these up. It's your keywords of coarse focus, the mirror, and the fine focus. And I will have another drink. Okay, so again, the language slightly different, but that's fine. We're saying the same thing here. So the coarse focus, the mirror, and the fine focus. Are you ready to see how you did? Okay, the coarse focus moves the stage up and down. It moves it up and down very fast. The mirror reflects the light onto the stage. Okay, so we went through that. And the fine focus is used to make the image clearer. So like I said before, the fine focus, once you've found that initial object that you're looking for using the coarse focus, from going up and down, moving down slowly until you find what you're looking for, you use the fine focus because it's much finer, it's much slower, it's much smaller increments to make that image clearer. So you use the coarse focus and the fine focus together. Always start with the coarse focus and then the fine focus to really fine tune it. Okay, name the highlighted part of the microscope, please. What do we think? What do we think? What's this highlighted part here? There's a circle, blue circle around a lens. And it's not the top lens, it's the other one. It's the one closer to the object, which makes it the objective lens. Okay, that's the objective lens, the one we can change. So we can change the objective lens to change the magnification if we want to get a closer look. Okay, so what's this knob down the bottom? You're probably thinking now, I remember there was two of them. Something to do with focus. They're both to do with focus. What do you think? Coarse or fine? Of course it is. It's the fine focusing knob. It's the fine focusing knob. And this here, okay, it's underneath the stage. It's a reflective surface of the light, which makes it a mirror. Okay, that's our mirror. Oh, and here's the other one. Okay, so we've got our other focusing knob at the top, and that has to be the coarse focus. Yeah, so the coarse focusing knob, and that's the one that moves the stage up and down very fast and in larger movements than the fine focus. Okay, so we've done one of the lenses. You all agreed it was the objective lens, the one that was near the object. So what's this lens? What do you put up against it? You put your eye up against it, so it must be the eyepiece lens. Okay, good. All right. The big part of the microscope where you put your slide. This is where you put your specimen, whatever it is you want to be looking at. Remember, the theatre has one of these. And that's your stage. Okay, so you set the stage and you put something on it. Right, let's look at using the microscope then. We're going to look at this method for using the microscope. So when you're in school, for some reason, it's always about onions. Onions, onions, onions. Now, onions are great. The reason we use onions is because you can get a really clear look at what's going on inside their cells, which we're going to get onto in later lessons. So we're going to look at what's inside cells, and plant cells in this one. But for now, we're going to use the onion to demonstrate how to use the microscope. So we're going to look at 5 steps. Step 1, preparing your slide and placing it on the stage. So that would be the first thing you do, is you put something on your slide and then you place it on the stage. Otherwise you'd have nothing to look at. There's your slide. And what do we think the slide's made of? Jelly? It's got to be something you can see through clearly. And it's glass. So we use glass slides and glass little cover slips. So the cover slip is this thing here. And the slide itself is this long one. Okay, that's the slide. Whee! Terrible, terrible, terrible writing with a mouse. That's your slide, and the cover slip is underneath. Use the lowest magnification. That will be the second step. First step, prepare the slide, place it on the stage. Next step, use the lowest magnification. So we're using the lowest objective lens. There's a few to choose from. It depends on the microscope, but always pick your lowest magnification to start with. Otherwise, it'll be very difficult to find anything. Okay, you zoom past it, or don't use the word zoom, you fly past it. And this is an example of a few objective lenses here, okay, on a microscope. The lowest magnification here would be this smaller one at the back here, and then you would work your way around. Oops. So the lowest magnification in that case would be a 4 times. Move the stage closer to the lens. This is step number 3. Okay, so we've set the stage, prepared our slide, set it to the lowest magnification, and then we move the stage closer to the lens. Okay, so we're going to move the stage closer to the lens, as close as you can get it. You wouldn't be able to see anything necessarily at this point, but it's your starting point. Okay, so you would look to the side of the microscope, just like in this diagram. You would look at the side, and you would get the slide as close to the objective lens as possible, and then you would start to move it downwards. We'll get to that. So by turning the coarse focus, you move the stage as close as possible to the objective lens. Your turn. Right, task. Put the following in the correct order. These are the 3 steps we've looked at so far. This is how we start to use the microscope. I'd like you to have a go at this, please. You can do this in your head if you want. You can write it down. It's all good. Okay, off you go. I'll give you a minute for that as well. Okay, how did we do? Get ready to see how close you were. So we're preparing the slide first. Of course we are. We need something to look at. So prepare your slide and place it on the stage. Step 1. Then you select the lowest objective lens. Okay, the lowest objective lens, that's the lowest magnification. In the previous example, it was 4 times. And then adjust the coarse focus to move the stage closer to the objective lens. Okay, so you adjust the coarse focus to move the stage close to the objective lens. That's your starting point. You are now ready to go on to step number 4. So step 4 is looking down the eyepiece. Okay, so first of all, we want to look down this eyepiece. We've moved the stage as close to the objective lens as possible, and now you're going to look down the eyepiece. You're going to slowly turn that coarse wheel to move the stage away from the objective lens. So now the stage is going to move away from the objective lens. It's going to start going down until the cells come into some kind of focus. Okay, so you're going to find these cells. Okay, and once they're in focus, you know you're in the right plane, the right part to be looking at this. Step 5, you try a higher magnification. Okay, so once you've found your cells, you change the magnification so you can get a closer look at these cells. Okay, because when you're on a lower magnification, the cells will still be very small. The screen will be full of cells. You'll change it to a higher magnification, and then you'll be closer. You'll probably see only a few cells in the eyepiece. And this is where your fine focus wheel comes into play. So turn to a higher objective lens and use the fine focus to get a clear image of the cells. So now that you've found your cells, you've increased the magnification. So you found your cells using the coarse focus. You've increased the magnification by changing the objective lens. And you can use that fine focus now to get a real clear image of those cells. And this is what it should look like once you get to that point. Okay, so this is on a higher magnification, and you can see each of those onion cells. That's your onion, by the way. Those are the onion cells. Okay, what I'd like you to do now is identify the mistakes in this method. So someone has written a method here. It wasn't me, promise. But someone's written a method and it's not quite right. So each one of these steps has got one thing wrong. Okay, so I want you to read through these, identify the mistakes, and we'll have a look to see how well you did in a minute, okay? So just read through them first, or take one at a time if you like, identify what's wrong with it, and then correct it in your head or write it down. I'll give you a few minutes for that, okay? 3 or 4 minutes for that. For those that have started, for example, number 1, prepare onion cells on a plastic slide. Do we think it's a plastic slide? I'm not so sure it's meant to be plastic. Perhaps it's another material. Number 2, select the highest magnification on the objective lens. The highest magnification. Not so sure that's sensible. Okay. A high magnification will make it very difficult for you to find what you're looking for. You want to start a little bit further out than that. Okay, 30 more seconds. Okay, so let's move on. I want you to have a look at the answers now coming up on the next slide, and oops, mark your own work. See how you're doing. Prepare onion cells on a plastic slide now. Of course, it's a glass slide. Okay, so it's always a glass slide. Select the highest magnification on the objective lens. Don't want the highest. Definitely don't want the highest. So we start with the lowest magnification. Okay, we're making life easy on ourselves here. Move the stage closer to the objective lens. Yep, that's right. Using the fine focus knob. No, the coarse focus knob. Okay, so if you were using the fine focus knob, you'd be there all day because it makes the stage move so slowly, it would take forever for you to get it up to the objective lens. So you move it closer using the coarse focus knob. Then you look down the nose. No, who made this? Me, of course. You look down the eyepiece lens, okay? You look down the eyepiece lens. You don't use your nose to look down a microscope, okay? So use your eyepiece lens, look down at the specimen, and slowly move the stage away from the objective lens. So now we're moving the stage away from the objective lens. And finally, rotate the lens. If I just move me out of the way so you can see that. Rotate the objective lens to a higher magnification. Yep. And use the fine focus, not the coarse focus, to make the image clearer. So at this point, we use the fine focus. Once we've found what we're looking for, we use the fine focus to make the image clearer. Okay, good. All right, let's look at magnification. I'm going to go back in the corner. Let's look at magnification. How do we calculate this? So can you remember the magnification of the eyepiece? The eyepiece is 10 times. So an overall magnification is equal to your eyepiece lens multiplied by the objective lens. Okay, so the two lenses on the microscope, the eyepiece lens, the one you look down, and the objective lens, the one which is closest to the object. Okay, you multiply those two together and that gives you an overall magnification. Okay, that will tell you how much you've magnified the image by. So if an eyepiece lens is 10 times magnification, and a student had the objective lens set to 4 times magnification, what is the total magnification of the image? So this is an example we'll do together. So you would multiply the two together, and that would give you a 40 times magnification. So you've taken it from 10 times magnification and 4 times magnification independently. Together, they are 40 times magnification. Okay, calculate the magnification if a student uses an eyepiece lens of 10 times magnification and an objective lens of 5 times. So one lens is 10 times and the other is 5 times magnification. What do we have to do to get the overall magnification here? The two lenses, they need to be multiplied together. Oops, excuse me. So the answer would be, in this case, 5 times 10, which would give us 50 times magnification. All right, have a go at this one. Calculate the magnification if a student uses an eyepiece with 20 times magnification and an objective lens of 2 times magnification. Okay, fairly straightforward, hopefully. 20 times 2 or 2 times 20. Okay, and you've got 40 times magnification. Finally, what about this one? Calculate the magnification if a student uses an eyepiece with 10 times and an objective lens of 35 times magnification. So you've got 2 different magnifications, 2 different lenses working together. What do we have to do to those numbers to get our overall magnification? Yeah, we have to multiply them together. 10 times 35, stick a 0 on the end. You've got 350 times magnification. All right, that's calculating magnification. Eyepiece times objective lens. What I'd like you to do now is have a go at doing those ones on your own, okay? So we've done the top one again. 10 times 10 gives you 100 times magnification. Remember to go through the process. Eyepiece lens multiplied by the objective lens. That will give you your total magnification. I'll just give you a minute on that. Hopefully you can do that in your head. Maybe need a calculator. Should be fine. Okay, so 10 times 40 should have given you 400 total magnification. 10 times 100 should have given you 1,000 times total magnification, and 20 times 50 should also give you 1,000 times magnification, because 10 times 50 is 500. Times that by 2, you have 1,000 times magnification. Okay. Again, let me know how you're doing in the comments, guys. If you're flying high right now, shout about it. Calculating image size. So we've done magnification. This is the only other thing we want to calculate. So now calculating the image size. So the image size is what you're looking at when you look down the eyepiece lens. When you look down your eyepiece lens, you are looking at an image, and that has a size, but it's not the actual size. Okay, that'd be crazy if the cells were the actual size of the image size. That would make the onion from earlier the size of London or something. So the image size is equal to the actual size times the magnification. So we've worked out the magnification. We can do that. The actual size is simply knowing what the size of the object on the slide is, the specimen is. Okay, sometimes you can do that measuring it using a ruler. So for example, here we've got what looks like a bee. Okay, I was hoping it was a fly, but I've just noticed it's got stripes on it, so it may be a bee. There's an actual size, and then the image size would look much, much larger. So if an object is 5 millimeters long, so you can use a ruler, you measure your object, it's 5 millimeters, you know that for sure. But you magnify it by 10 times, so you've got 10 times magnification. How big will the image make it appear? So we know the actual size is 5 millimeters. We've magnified it by 10 times. How big will the image appear? Yep, so it's 5 millimeters times the 10 times magnification, and that would give you 50 millimeters. That would be the image size. Okay, so that's like the picture there on the right. You can see the actual size. Let's say that was a 5 millimeter long bee fly, and when you've magnified it 10 times, you end up with an image size of 50 millimeters. Right. I'd like you to have a go at this one. Calculate the image size of a 2 millimeter piece of apple. So we cut off a 2 millimeter piece of apple under a 10 times objective lens. Okay. 10 seconds. 7. 5, 4, 3, 2, 1. Okay, good. So it's 2 millimeters. We need to multiply the actual size, 2 millimeters, by the magnification, which is 10 times, and that would give you 20 millimeters. 20 millimeters would be the image size. Newspaper. Calculate the image size of a 3 millimeter piece of newspaper under a 100 times objective lens. Okay, so it's 3 millimeters. Measured it, definitely 3 millimeters. Put it on a slide. Put it up against the objective lens. Use the coarse focus. Use the fine focus, and then we've got our image. What is our image size? It's 3 times 100, which gives us 300 millimeters. Calculate the image size of a 5 millimeter piece of onion under a 50 times objective lens. Okay, here we go again. So we're multiplying together. So we've got our actual size of 5 millimeters. We're multiplying this piece of onion by 50 times magnification. So 5 multiplied by 50. What does that give us? So that should be 250 millimeters. Okay, so we've got a 250 millimeter actual size image. Okay, calculating image size. So calculate the image size in the following questions. I would like you to have a go at A and B and push yourself if you can to do the challenge. The challenge will take a little bit of working out magnification, but we've already done that. So if you can remember how we calculated magnification, you should be able to have a go at the challenge. Right, I'll give you a couple of minutes to have a look at that. Okay, so A and B are, again, the multiplication of the magnification by the actual size. And for the challenge, you need to calculate the magnification using the eyepiece and the objective lenses before you can do the overall calculation. So once you've got the magnification from the eyepiece and objective lens, you can put that into your equation to calculate the image size. Okay, so if a student uses a microscope which magnifies 200 times by 10 millimeter, a 10 millimeter piece of onion, you have to do your calculation of the magnification times the actual size, so 200 times 10 millimeters. Oops, sorry. And that would give you 2,000 millimeters. A 2.5 millimeter piece of flour is placed under a microscope which magnifies 40 times. So you've got 2.5 millimeters times 40. So your magnification 40 times your actual size 2.5. And that should give you, drum roll, 100 millimeters. And for the challenge, you have to calculate the magnification. So you need to multiply the eyepiece lens by the objective lens first. So 10 multiplied by 10 gives you 100 times magnification. Now you know 100 times magnification times your actual size of 5 millimeters. This piece of hair, what's the image size going to be? It's going to be 500 millimeters. Okay, well done if you got that far and you worked that out. And there's your answers. Let's pop it there and there. Okay, so how do we do? Did many of you get onto the challenge? Did you understand the challenge to try and calculate the magnification before you could do the equation for image size? Okay. Right, our final task, okay? You're doing well. There'll be a lot more microscopes further down the line, okay? And they'll be looking closer at how to prepare a slide and much more interesting things of looking inside cells, getting a closer look at objects. But for now, I'd like us to finish off with putting these following steps in the correct order, okay? So if you can do this, you can give yourself a massive pat on the back, because this is the aim of today's lesson, to be able to put these steps in the correct order of how to operate a microscope. So read through them carefully, and you don't have to write out the sentences. If you've got a pen and paper or pencil and paper, just write the letters in order, okay? So if you think, for example, and this is a massive hint, if you think F is the first one, write F as your first letter, followed by the next step. So F, put the slide on the stage. That is a really good starting point. I'm going to give you 2 to 3 minutes to write out your letters read through this, write out your letters, and then we're going to see how you did in a couple of minutes. Okay, thank you. Okay, just 30 more seconds on that, and then we'll go over the answers. Right, okay, let's do it. There you go, guys. F, C, A, D, B, and E should be your order. So put the slide onto the stage. Then next turn the objective lens to the lowest magnification. Okay, very important. A, look down the eyepiece lens. So now we know we're on the lowest magnification, we can look down the lens. Then we start to move the coarse focus knob, okay, to find some cells. Move the slide close to the objective lens and then turn that coarse focus knob to find some cells. Once you've found some cells, you can increase the magnification by turning the objective lens to a higher magnification, okay, to view the cells in more detail. And now we've magnified those cells, we need to turn the fine focusing knob to bring those cells into focus, okay, because they won't be in focus. You found them, you found the correct plane, we call it, okay, you're in the right area, but you've got to turn the fine focusing knob now to bring those cells into sharper focus. Okay, well done if you got all those right. Okay, that was the main aim of today's lesson. If you'd like to leave some feedback in the comments about how you found that, did you get 6 out of 6? Did you get 5 out of 6? How close were you? Was there anything that that's confusing you in some way? That you're getting two things mixed up, perhaps? Okay, anything like that. If you leave any comments at all, I will respond to them. We will be going over microscopes. We'll be revisiting microscopes when we're talking about other topics such as cells and plant cells. And yeah, we'll just keep revisiting this. This will pop up time and time again, and this is a good introduction to it at this point. I'm going to leave you with this, okay? So we'll finish the lesson there. If you'd like to have a go at this next question, can you describe a method of how to prepare and look at a cheek cell? So this would be really stretching you. I'd like you to imagine now you've got a microscope, you've got a cheek cell, okay? You've got some cheek cells that you've scraped out of your mouth. How would you prepare those cheek cells? Okay. Now, considering you probably didn't know much about microscopes before this lesson, it'd be interesting to see if you could have a go at this in any way, okay? What do you think you would do? You've got everything you need. You've got your glass slide, you've got your microscope, you've got all the parts of the microscope that we've discussed. You've got your two lenses, your eyepiece lens, your objective lens, your coarse focus, and your fine focus. And the different magnifications of the objective lens. Do we start with a high magnification or a low magnification? Where do we put the glass slide? Does it go underneath the microscope? Does it go next to the microscope? Does it sit on the stage? Okay, so have a think about that. Write down some bullet points and we'll see how accurate you can be. Give as much detail as you can. Just bullet point it, no paragraphs, not looking for an essay. Just a few pointers about how we've been talking with the starting with putting the slide on the stage. Setting your magnification. Do you want a high magnification or a low magnification to start with? Do you adjust the coarse focus knob next? Or the fine focus knob? Which one do you need to adjust to find your cells? Should you be looking down the eyepiece lens at this point? Or are you just going to guess? You're just going to turn the coarse focus knob, hoping? When you found those cells, what are you going to do to the magnification? Okay, because you're quite far away from the cells. They look quite small, okay? They're quite small when you look down the eyepiece lens. You found them, they're quite small, so you need to do something to the objective lens. And then, of course, once you've magnified them further by changing that objective lens, which focus wheel do you need to turn next? How do you get them sharp? How do you get them really in focus? Do we use the coarse focus or the fine focus wheel? Okay, just one more minute, finish writing off your notes, check over them, see if you're happy with the order. Do you think you could do this now if I gave you a microscope and the slide? Do you think you could instruct someone else to do it? Okay. Something like this. It doesn't have to be exactly word for word, right? But something like this. Prepare the cheek cell and place onto a glass slide and clip onto the stage. So get that cheek cell onto the glass slide. Select the lowest magnification on the objective lens. So yes, we want the lowest magnification, not the highest. Look down the eyepiece lens. Okay, so look down the eyepiece so you know what you're looking at. Move the slide close to the objective lens by turning that coarse focus knob, and increase the magnification by changing the objective lens. So move the slide closer to the objective lens by turning the coarse focus knob until you find both cells, and then increase the magnification by changing the objective lens to view the cells in more detail. Okay, so that increases the magnification. You get a closer look, you can see more detail. And then turn the fine focusing knob to make the image clearer. And that is all I was looking for. So anything like that, anything close to that, well done. Well done at this point. That's really good. Okay, so we'll leave the lesson there now. Hopefully that will sink in. You can go back, you can revisit this lesson. It's going to be here for you to look at as much as you want to have a look at it. And yeah, leave any comments you want, any questions you've got. I'll get back to you and try and answer as many as possible. Okay.